We're here at the AAPS National Biotech Conference with Michael Anderson of ICON plc. He is the Bioanalytical Services Manager for the EU. Michael, welcome.
Thank you, Elizabeth.
To start off, can you tell me what the top emerging trends are in large molecule bioanalysis?
Sure. I think there's a handful of emerging trends which we're seeing a lot more focus at this meeting. The first of which is antibody-drug conjugates and the unique bioanalytical aspects that go into developing a bioanalytical program around these molecules, specifically around the use of three independent PK assays, one for the antibody conjugate as a whole, and one for the antibody itself, both by immunoassay, and then an assay for the conjugate itself by LC-MS. So with the recent publication of a lot of clinical trial data that's shown the pretty effective use of ADCs in treating cancer, we think this is going to be a fairly high-profile emerging trend over the next couple of years. Another pretty high-profile emerging trend is biosimilars. So particularly around the debate of 1 versus 2 assays for PK and ADA. So in the PK assay, can you truly compare, or can you truly prove comparability when you're using 2 or 3 reference standards in the assay? And a more complicated topic is the choice of whether to use 1 or 2 assays for your immunogenicity program. A third emerging trend is really large molecule by mass spec. So this is something we're seeing a lot of both at this conference and as a CRO in the industry. It's requiring quite a bit of collaboration between our mass spec scientists and our immunoassay scientists. So whether the molecule is a peptide or a large protein that requires digestion, there's typically an antibody-based immunoaffinity precipitation step that precedes the analysis. So we're of the opinion that this is going to be a fairly hot and emerging topic over the next couple of years.
And speaking of the National Biotech Conference, how is ICON plc involved in the program this year?
So we're pretty involved with this year's program. I myself am on the AAPS Action Program Committee for Biosimilars. We have a hot topic session at this meeting Wednesday afternoon, and we've been tasked with publishing an industry white paper to provide guidance on PK and ADA validations when developing a biosimilar. So ICON as a company is very involved in biosimilars, and we feel that gives us a fairly unique position to contribute to the Action Program Committee. We've developed and validated internal methods, both PK and ADA and neutralizing antibody, for a variety of molecules which people are developing biosimilars against, the likes of rituximab, infliximab, etanercept, and trastuzumab, just to name a few. So based on that, we feel we're well positioned to contribute to the industry. And ICON recently did a webinar to talk about the bioanalysis of biosimilar programs, and particularly around whether to use 1 or 2 assays, the information of which can be found on our website.
And there seems to be continued focus on immunogenicity. How does ICON support ADA assessment?
So certainly there's a very, very heavy increase, both regulatory and industry-wide, on immunogenicity, especially as protein therapeutics move into the more risky category. So it's also one of the core competencies and core expertise areas of ICON's global immunoassay service offering. So we have extensive expertise in a large number of protein biotherapeutics that require an enhanced focus on immunogenicity testing. We also have extensive expertise on working in inflammatory autoimmune disease states and oncology disease states, both of which require fairly unique aspects when you're setting up your immunogenicity program. ICON also excels in working with our sponsors up front when we're working with inflammatory autoimmune disease populations to deal with pre-existing anti-drug antibodies, which is probably another one of the emerging trends, but it's a continued focus at the National Biotechnology Conference and other meetings. And ICON can provide unique and innovative solutions to deal with pre-existing anti-drug antibodies, so your data is presented to your regulators in the most appropriate fashion.
Thanks, Michael. To find out more, visit iconplc.com.